Okay. Let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all, and welcome and thank you, SynBYSS people, for joining us, the 101st seminar, and I call it on Imperial College London Day. In most northern hemisphere regions, the temperature has dropped significantly after very, very hot summer, showing some colorful leaves and fall foliage. I should go hiking with my wife this weekend, probably after finishing my paper revision for submission and some presentation preparation. But I guess life is short, fall is even shorter. So let's enjoy in this beautiful weather. It is also a bit longer than 1 month since my last trip, and I look forward to embarking on my trips to Washington, D.C., Germany, and Orlando to attend conferences and EBRC council meeting, catch up with colleagues, and meet new people. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Karen Polizzi. She is a professor of biotechnology, Department of Chemical Engineering and Imperial College Center for Synthetic Biology at Imperial College London. I believe I met her for the first time at Synthetic Biology Gordon Research Conference many years ago, and recently at Imperial College London last year, and also Cancun for the conference last year. And when I gave an invited seminar there, and more recently again at the Synthetic Biology GRC this year. She is one of the leaders in synthetic biology, doing fantastic research, and importantly, she will be the chair of Synthetic Biology GRC in 2025, one of my favorite conferences. So Karen, thank you so much for your leadership and service in this field, and also your dedication to education of the next generations. And the virtual podium is all yours now. And thank you again.
Great, thank you so much. Let me share my screen, and thank you for having me. You'll see hopefully the slides and also in projection mode. Okay,
yes,
perfect. And then I will just in case get the pointer ready. So I'm really honored to be here. And when you get to the end of my talk, you'll see what I mean by the following statement. I resisted quite a long time coming to this as a pioneer speaker because I thought the word pioneer wouldn't apply to me. And so one of the things I want to address today is imposter syndrome, which I think many of very successful people in science have. And so if you're not aware that it's a thing, you might actually feel afterwards that this could also describe you. But I promised I would also talk about science. So today I'm going to just briefly go through some of the more recent work in my lab, which generally applies synthetic biology to bioprocess engineering. We kind of have two different streams that we work on. So we do cell-based and cell-free biomanufacturing of proteins, various therapeutic proteins and vaccine targets. And we also develop new analytical technologies and biosensors to help with bioprocess monitoring or identifying bottlenecks. So if I speak about the biomanufacturing part first, essentially what we're doing is just explaining the fact that cells can already make a lot of really complex products and just coaching them to do things in a more reliable and robust manner for our own purposes. So when I started my group, we did a lot of work in therapeutic protein production and Chinese hamster ovary cells. This is a mammalian-based platform. And we were making a lot of protein-based drugs, but they also have sugars attached to them. The glycosylation process, it's very non-templated and it can be affected by the process conditions. So we were trying very hard to understand how we can follow what we're doing in the bioprocess all the way through to the end product, and to manipulate the conditions to control product quality and purity. More recently, we've been working on vaccine production in a yeast called Pichia pastoris. So this is a methylotrophic yeast. And because it's a microbe and eukaryotic, it has a lot of the complex features that mammalian cells have, but it's a lot more simpler to engineer. And if you're thinking about a cost-effective process, for example, for vaccine production, it's also a much cheaper host to work with. And one of the reasons we like Pichia is that it's got high cell densities that it can grow to, and therefore you get high product titers in the culture medium. And we've been working a lot on tools to develop high-yielding strains. So it is a yeast, but it's not the same as everyone's favorite yeast, Saccharomyces cerevisiae. So there are some challenges. And a lot of this work has been in collaboration with the Future Vaccine Manufacturing Research Hub, which is a large initiative that we have at Imperial College, looking at different non-mammalian vaccine platforms. And I'm honored to say that we were given the ICANNI Global Award in 2021, along with the Vaccine Hub at UCL, for our contributions here. Also in biomanufacturing, we're interested in cell-free protein synthesis. So this is essentially the contents of the cell without the cell wall. It makes it a lot easier to manipulate the reaction system. And therefore, if you have difficult to express proteins, sometimes it works very well. And it also has been shown to scale up very well by the industry leaders in this area. For our purposes, we've been working with both Pichia and Chinese hamster ovary cell-free extracts. And we want to use this as a prototyping system. But what is critical for this is to maintain the post-translational modifications that you would see in the native whole cell hosts. And so we've worked a lot on developing a system for in vitro glycosylation, where you can manipulate the glycoform towards your desired structure. And this work is just in revision now, but you can check it out on bioRxiv. In the second half of the lab, we're working on these new analytical technologies to support this process. And one of the things that we look at are genetically encoded biosensors, specifically for small molecule metabolites. And the reason we became interested in this is because there are many, many small molecule metabolites in a cell, and we need to be able to monitor these during production and figure out where there might be bottlenecks. And this would allow us then to develop bioprocessing solutions or to decide what kinds of genetic modifications might be helpful to improve productivity. And what's obviously essential for this is to have sensitive, specific, and quantitative responses. So we've done a lot of work on how you take GFP-based reporters and make things more quantitative. But then I really had an issue getting people to accept that they might want to use these kinds of new analytical technologies in a bioreactor. So we've started to think now about practical solutions here. So how best can you deploy these sensors in a manufacturing context? And really with a focus on getting something that manufacturers would adopt. And so some work in this area has been to encapsulate E. coli-based biosensors inside polymers, and then you can co-culture them with mammalian cells without there being any impact on the mammalian productivity. And also this work, which was started by some PhD students, where you can look at basically a whole cell version of the agglutination reaction that gives you a very quick and simple readout that's compatible with diagnostic workflows. But I began the talk talking about the imposter syndrome, and really I think it's the most inspiring thing that I can do, since Tae-suk's brief said, please say something inspiring. If you've not heard of this, I guarantee you that you will recognize it in yourself. So imposter syndrome is when you feel your success or achievements are not deserved, or you feel inadequate, or you feel that others will discover this about you. And hopefully I controlled my facial expressions really well, but when I was being introduced, it was really, really hard to not belie what's going on inside me, thinking, is that really me? Is that how others perceive me? This is very common in a lot of fields, especially where people are high achievers. And so as a consequence, in academia, it's incredibly prevalent. In some studies, as many as 70% of respondents identified with these feelings. And actually, if you start to have conversations with people you know, some of the most successful people I know at Imperial College or that I've worked with abroad have these kinds of feelings. And so it's a very, very common thing. The reason I want to bring it up is because it is a barrier to diversity. It's much more common amongst early career researchers, amongst women, amongst people who are in their first generation to go to school, amongst those who are the first to get the PhD in their family, and those with other protected characteristics. So I think it's something that we as a community need to watch out for if we want to help these people to get over this, put themselves forward for new positions and academic positions and things like that. And I really think that the first step in fighting it is to recognize it. So first of all, in yourself, but also maybe in those that you are mentoring. And if you can each tell 3 people about this who don't know about it, I'll feel like the word will spread, and this seminar has really been useful. So with that, I will end. I want to say a special thanks to all the group members, past and present, and the funding sources, of course, because without them, we wouldn't be able to do the work. Thank you.
So wonderful. Thank you so much. This is amazing and brilliant. So I agree with you about the syndrome you just mentioned. And I believe, many of us just constantly comparing us to others. And that's probably one of the reasons or cause. But I believe we cannot compare ourselves with others because we are different. And the most important thing is we are different. And as long as we are happy with what we did, I believe that's the most important thing rather than publishing 10 Nature paper in 1 year, right? So, and everyone has different role or different task or mission in this world. And as long as we achieve that goal, I believe we living the right, satisfactory manner, I guess. So that's wonderful. I mean, thank you for bringing up. That's one of the important issues in academia. I 100% agree with you. Thank you. Wonderful. All right, so let's move on. Okay, now the main speaker of today with a longer introduction. Dr. Lin Su is a Leverhulme Early Career Fellow at the University of Cambridge. He graduated from Southeast University in 2021 with a major in biomedical engineering. He studied synthetic biology while visiting Caroline Ajo Franklin's group during his PhD. This research focused on engineering electron transfer at the interface of microorganisms and materials. Applications of his research include building biohybrid systems to perform semi-artificial photosynthesis and designing bioelectronic sensors for information exchanges. Importantly, he is one of the conference committee members helping organize the first SynBYSS in-person conference in Hawaii next year. The details will be announced soon in social media or by email, so stay tuned. Lin, thanks so much for your time today and for helping me organize the conference for this webinar series, but now in-person, conference in Hawaii. So why not? Let's attend all of us. And thank you again, and please, and take it away, and all yours now.
Thank you, Taesuk. That's very kind introduction. Let me try to figure out share my screen, if you can see that.
Yes.
Great. Well, again, thank you for having me here, and also thank you for organizing not online ones, but also trying to organize the in-person ones, because we do know that's take a lot of effort. So again, I'm Lin Su. So I'm currently a postdoc research fellow in Cambridge, but today my talk is mainly present the work I did during my PhD, especially my visiting at Ajo Franklin's lab. So the title will be Real-Time Environmental Monitoring of Contaminants Using Living Electronic Sensors. So basically, idea is just trying to use engineer or synthetic biology to engineer microbials and sending out electrical signals for sensing. So let's move on. So before we jump into the science, let's talk about something different or a simple question. So right now I'm presenting a map of part of the Cambridge University. So we have the river Cam just going through the city. And now a simple question. What if I need to move something to across the river? So obviously there's several easy ways, maybe just already come to your mind. So for example, you can build a bridge. Or if you enjoy punting, or we call it punting here, but basically if you can get a boat, you can just use the boat to help you to cross the river. And of course, if you are more confident with the scales, you can definitely swim through the river. So that will be how you're going to cross or move things around a river. Now let's change the topic a little bit. So imagine you have a cell. And now you have a river of lipids. Well, we know the cell membranes are made by lipids and some of the proteins decorate on that. And it's kind of helping us or helping the cells to maintain a nice environment in both pH and other reaction nutritions, and also defect whatever the effects from outside. But now if I'm asking you, if you can think some ways to move electrons across this lipid river, how are you going to do that? And of course, that's the first question. But second, probably you're just going to ask me or ask yourself already, like, why you bothered to do that? So I hope by the end of this talk, I can kind of convince you not only how we can do that scientifically, but also what's the purpose we bothered to think about this kind of question. So let's go to some basic science about how we're going to move electrons. So we all take food. Oh no, I know, we know UK is not particularly proud of the food here, but the fish and chips still delicious, so I highly recommend. But basically, when you're taking food or consuming energies or generating energies, if you look at the formula here, so basically, for example, what we learned from our textbook is when you have glucose and you have oxygen, and then you can convert it to CO2 and water, and also you're generating energy. And if you look a little bit detail, like how you generate ATP or the energy, you have electron transfer chain, maybe on the cell membranes for the bacteria, or maybe mitochondria for human cells. But again, you will have a thing we call electron transfer chain and moving electrons around, and also the protons. The protons then will eventually just help you to synthesize ATP. But today we want to look at how the electrons or how the oxygen take part inside of the reaction. So if you look at the middle of the image here, so you have two protons and some electrons, which are not shown here, but also oxygen, and you have the reaction to generate water. The oxygen here, well, basically that's the reason why we need to keep breathe. But also, if in the field of redox chemistry or electrochemistry, we basically call oxygen as an electron acceptor. And the reason we call it just because they have reactions to neutralize or take the electrons and convert into water. And now, if you look at environment that there's not a lot of oxygen available, how the lives there are going to make energies or make ATPs. Of course, you can think about fermentation and other things, but there's also some unique pathways. Some of the bacteria, they can develop during the evolution. So they can form either like conductive nanowire or some cytochrome proteins on the membranes, or even they just have some small molecules as a shuttle to not taking electrons, not taking oxygens, get inside of cell and finish the reaction because they do not have oxygen available. But instead, they have these different mechanisms to move electrons from the inside to the outside and just throw the electron out and finish the reactions outside, for example, with some mineral oxide or metal oxide, this kind of thing. And we call, or the field call this phenomena, we call it extracellular electron transfer. Basically, you get ideas like we're trying to get electrons to extracellular and then finish the reaction by this kind of electron transfer. So that will be the core science behind today's my talk, and most likely when we're talking about how you're engineering the electron transfer or electron connection between the living and the non-living system. So the living system, which just refers to the bacteria in this talk, and non-living systems could be some other materials or electrodes. So now we know how the electrons can be moved across the membranes, and then I'm trying to help you answer the second question, like why. So in this field, because it refers to electron transfer, so we have been developed applications like could be the electrons used as energy. So we have developed a microbiofuel cell systems that you can just hopefully just plug into a bacteria and power your phone and power something else. And also because it's an electron transfer, and you have redox reaction happens during the process. For the people works through the waste treatment, they usually use this as the energy, as a cheap energy input, trying to neutralize some of the pollutants, because you can just easily just reduce them without getting extra cost. And of course, because you think we're talking about electron transfer, and it has to be dual ways. Like you cannot only thinking about electrons going inside the cell to the outside, but also you can do the reverse. So there's also studies like how you use the electrons to power bacteria and to do electrosynthesis, to synthesize useful chemicals. So of course, we are in the same of the synthetic biology. So in 2021, we do write a review about what we can do as a synthetic biologist to contribute to this field. So obviously you can say, okay, we do engineering, we can increase the power output. We can also kind of do metabolic engineering. So when you pump electrons inside, you can have your customized synthesized to get whatever the compounds you want to. And also the other area we're trying to say is, okay, it's electrons. Because nowadays, even at the meantime, you guys can see me or hear from me just because electrons are carrying the information. So we also think about, okay, if we can engineering the bacteria and kind of use it for sensing, or even more advanced, like some circuit or electronics, we can do computing based on the electron transfer. So basically, we were thinking the electrons not only a carrier of the energy, but it could possibly use as carrier of the information. So now bring us to the sensing, which is the core topic of today. So if you compare biosensing or biosensing with electroactive bacteria, which is just the bacteria I referred, they can do electron transfer. The big difference here will be how you convert the signal. So traditional biosensing, you can imagine you have a cell or part of the cell component. You can just have GFP, or you can have pigment, you have enzyme to catalyze reactions, and then you can detect the GFP signal, you can detect whatever the colorful pigment you come out, or you can detect what's the product or the consume of the substrate from the enzymatic reactions. But to convert it into information that extremely, if you can read from your computer, from a machine, you need to have a transistor to convert all different kinds of signals in fluorescence, in color, etc., and into electrical signals. But if you use the electron transfer-based biosensing, you don't get rid of the transistor, because whatever output from this kind of bacteria is already an electrical signal. So which means you simplify the system or simplify the process, which can help you to make minimize your system, and hopefully you can save some money with the cost. So there's a two state-of-art or my favorite examples of using this kind of electron transfer and building biosensors. So basically you can, in the top, you can have some engineered bacteria that can respond to a chemical TMAO. And once the TMAO exists in the system, it turns on gene expression. The genes are essential ones in the electron transfer pathway. And that means when you have the chemical, you induce the gene expression, and then you can read out the electron signal because you have the essential proteins get expressed. Similar, in this case, if you want to sense not a compound or a complex chemical, but just element. You can also have a promoter that can respond to that, and then you can kind of have genes expressed related to electron transfer, and then you also read signal. So all those are very good examples to show how you can convert electron transfer into a biosensor. But they do have limits. One of the limits we're going to talk about today is the time. Because if you think about building a sensor, and the sensor is rely on gene expression, so basically means you need to have a protein being go through the central dogma. You have DNA to RNA to protein, and you need to time to cost in the transcription and the translation. So I'm just giving a quick example here, maybe not very accurate, but just trying to give you some ideas. So in the slides, I'm showing MTR complex, which is basically the key genes that in previously the studies used for the electron transfer sensor. And it has 3 different proteins, and in total, it basically have around 5,000 nucleotides, or in another way, roughly 1,700 amino acids. And if you consider you are using basically E. coli, one of the chassis we are familiar with, that they basically can do the transcription and translation very fast. But if you do the calculation, you can still easily take longer than 10 minutes for them, just asking them to convert the DNA into the protein. Well, 10 minutes sounds like it's not that terrible, but if you think about mutation or post-transcription modification, that will take extra time. So the proteins we mentioned in the examples, they are cytochrome proteins. 2 of them are cytochrome C, which means you need to insert heme groups into the cytochrome to make it functional. And during the process of mutation, you basically need to go through dozens of proteins and help you to fold correctly and insert heme group correctly. And that will take time. But unfortunately, I don't have the specific time like tell you how long it will take for mutate cytochrome, because it depends on different organisms and different protein, that time will be varies. But I can give you another example if you look at the fluorescence proteins. So in this case, I'm just showing if you want to mature the fluorescence protein, well, of course, you have different color options. You have blue, you have green, you have red, you have different ones, and in different chassis. And if you look at the time for the mutation, it still take a lot of time. Luckily, if you're lucky, you choose the right one, make it take minutes, but most likely in the case will take an hour. And if you combine all things together, and if also you take into account when you do the real sensing, you need to consider mass transfer limit. You need to consider if you get enough signal to differentiate from the background noise. Usually, this kind of biosensor will take hours to get you a reliable signal to tell the things you want to detect is rare. Now brings to today's question, or the main question, how we design a sensor and also they are fast. So luckily, when we start the research, we have a nice collaborator, Prof. Selberg. Prof's group is basically studying a protein switch or redox-related protein switch. The core idea is they can use protein engineering and separate a functional protein into 2 parts. And when you separate them into 2 parts, the protein is not functional because the two parts are separated away. But they also can put a ligand in between the 2 parts, and the ligand can respond to some of the things. For example, here, one of the chemical, FHT, which will be whatever we're going to talk in the following talks, will be our target to sense. And when the FHT is present in the environment, it reacts with ligand, cause changes of the structure of your protein switch, and the 2 parts of the separate proteins will be just fused together. And once again, it will become one functional protein which means then you can have some readout from it. And because Geoff is specifically looking at a redox-related protein, which means we can have electron transfer affinities across the switch turn on and turn off. So that's basically the way we are thinking when we start a project. We can probably use the protein switch to have a faster sensor, or to make it across a wide limit of the transcriptional, and the mutations time. Just because you basically have all the paths ready, what do you need to do? You just have you interest of chemical to bind to your ligand, cause the formation change, and then you can read out signals. And then how we do that? So we have been introduced a lot of electron transfer. So what Caroline A. Franklin's group, Michael's group, what they did, or what we did, is we partially just using synthetic biology tools to transfer some of the key genes. Again, the MTR pathways will be the same one I just showed before when we go through the calculation. So we express the key genes of electron transfer into E. coli and make E. coli can generate electron signals. Of course, I can show you more details later if you're interested in that, but basically we kind of figure out how to engineer E. coli and make it efficiently to send out signals or electron signals. So now we have a protein switch that we assume that can be acted fast, and also is related to electron transfer because the redox reaction of the enzyme. And also we have electron transfer pathway, which we can act as a electron signal output conduit. So now it's pretty straightforward, which as we're thinking or we're starting project, we just combine these two together with a protein switch to detect the compounds we want to sense, and we use the electron transfer pathway to deliver signals. So that's basically how we did it. So we have Josh Atkinson here. Back in the day, he was a graduate student in Geoff's group, and now he just recently become a professor at Princeton. So Josh helped us a lot with design and to make this complex electron transfer pathway. What we basically did is we have taken still the electron transfer pathway. Originally in Caroline's group, we engineered in E. coli, but also the pathways Josh designed that can also bind to a sulfur metabolic pathway in E. coli and transfer electrons. And the ideas we're trying to do that is basically we have a whole artificial electron transfer pathway, and we can use part of the pathway, or basically one of the proteins, ferredoxin here, which is previously engineered by Geoff's group into a protein switch. So we want to integrate the protein switch here, and we still have the O pathway coming out to send out signal, and we have some of the proteins in between to connect the electron transfer or help deliver the electron transfer. Again, if you think about how you cross or moving things across a river, across membranes. So I'm not going to show you more details about how we make the pathways or the electron transfer circuit. I'm basically just trying to quickly show you, because when we during the design, we're trying to split into 3 different modules. So we still have the output module, which is the electron transfer pathway. And you can see we can nicely demonstrate if you have the pathway and compared to no pathway, you have a big signal difference in electrons. So the current in the y-axis basically is what you're reading from the instrument or potential stat. We also engineered a coupling module, which essentially just an SQR protein that can connect with the sulfur metabolic pathway and also the electron transfer pathway, and you deliver electrons. So what SQR did is just basically taking 2 electrons from S2 minus and convert it to S0, and it takes the 2 electrons and to reduce the quinone. And later the quinone will just taking electrons and deliver to the output module that you're sending out electrons. And also, again, we have the input module, which eventually we're going to swap the ferredoxin in part of it and into the protein switch. And we use that as a signal inside of the cell for us to sensing. So we do verify these three modules, they can work separately. And the next, we're just putting things together. Where we're putting things together, we just give it to test if it's working. We just use it, the whole signal, whole electron circuit as a sensor for cell sulfate. So again, cell sulfate is not the target we're going to do for the sensing, but cell sulfate is also part of the electron transfer pathway if you look at how we design it. So we think that can be an easy test or faster test for us in the lab to say if our designed electron transfer pathway is functional. So the idea is if you put sulfur or cell sulfate in this case, it will be converted into S2-, then S0 through the SIR protein. And SIR, the reason SIR can do that is just because they're taking electrons from further ways in the metabolic pathway. And then SQR, I just introduced to you that can reduce quinone, and quinone can later just reduce the whole electron transfer pathway and sending out signal. And I'm not going to go to the details how we make that, but basically we do demonstrate if you spike different amount or different concentration of the cell sulfate, you can see a different electron response. You can see that. And if you just plot that and analyze it, it's quite linear with the concentration and the response. So we do confirm that, okay, this circuit kind of working. But now goes to the sensing, because that's the most important thing we're trying to explain to you how we make it fast. So now in this case, we just swap the functional ferredoxin into the ferredoxin switch that Josh made before. So again, a little bit recap, the ferredoxin switch will just respond to FHT, which is a chemical we want to detect, and the FHT will bind to the ligands and cause the changes of the two parts and get merged together as a full functional ferredoxin, and then hopefully you can still see the signal, and that's basically what we did. So in this experiment, if you look at panel C, when you spike DMSO, which will be the solvent for FHT, you put into that, into our design sensing system, and you basically see the output of the signal is quite flat, although it's kind of decayed a little bit, but still, if you have FHT that dissolve in DMSO and spike at the same time, you will see immediately you have a significant difference or significant signal output that you can vary from the background, which is the red color curve here. And if you do a little bit of statistics, you can say with 1 star or 95% guarantee, you can say in the time of 7.8 minutes, you can able to read out the signal of the FHT and to differentiate from the DMSO background. And of course, you can say you can probably take a little bit longer if you want to more confidence in the system. So now we already proved, okay, the protein switch is working and we can detect the chemical we want to sense in a reasonable short term, like less than 10 minutes. So if you still remember the calculation or the rough calculation, we're talking about the hours we need to have we need to take to ask the bacteria to express the functional protein and then get mutation successfully and deliver signal. 10 minutes is relatively faster than hours of time. And in the last bit of our work, we just demonstrate our sensing system can not only working in the lab condition, which is basically just some buffer we made, we're also taking samples from the real world. When we do the experiment, we will the whole lab was moved to Rice University. So we just take samples around Houston. So you have IU samples, you also have samples from the sea, which is the Galveston Beach. And we take the sample, which is a photo colleagues took for me. So we just go to the water source and take samples and we bring it back to the lab and we set up the experiment and trying to see if our sensor can respond correctly in different kind of water. Of course, we did a little bit more engineering, like we're using nanoparticles to increase signal or to decrease the detecting time. But eventually, we did not perfect, but some kind of demonstration with different kind of water system, and you can still detecting FHT, which again is the compounds we want to sense in this case, and in a relative short time. So hopefully, by far, I kind of convinced you that we do made a faster sensor, and a little bit of summary with the work is we kind of introduced a 3 different module and system, and it's for artificial electron signal transduction for detecting the chemicals. And also we successfully to implant the protein switch to enable us to do the sensing that fast rather than hours of waiting. And of course, we do take advantage of some engineering in the material side, like we use hydrogel and some conductive nanomaterials to increase the signal or help us to get a better sensing ability. So with that, I would like to thank all the collaborators or people's contribute in the research, especially Caroline helped me to get involved in the synthetic biology when I was in her lab. Moshe, who is the postdoc in the lab and start the ideas of this faster sensing. And Xu Zhang, she helped us when we doing the final demonstration in the sensing. And of course, Geoff and Josh, they developed the fastest sensing protein switch. And of course, they are very great collaborators in the world, basically. And also George involved in the discussion and help us to design the project. And also my professor supervisor back in the China Southeast University, he kind of introduced me to the world of electron transfer and also the nanomaterials we use. And of course, we're thanks to all the fundings that supports to the research. And a little bit one more thing. So since I mentioned the sensing work or the electron transfer is mainly done during my PhD, I do want to give a little bit of shout out to what we have been done during my postdoc in Cambridge. So still I'm studying the electron transfer as an interface. But now we're not thinking about information, we're thinking about energy. We're thinking about putting electrons inside the bacteria and to do synthesis, or we call that synthetic semi-artificial photosynthesis. So basic ideas is just trying to build a biohybrid system, not taking advantage of the synthetic biology, but also material engineering. So you have photosensitizer, this kind of a material response to the light, generating electrons, and you can transfer to bacteria. And in the field, we do have two of nice examples like I personally loved. So one is a science paper from Pei Dongyang's group. They have Morella semiacetica and a synthesized cadmium sulfide oxide of membranes, which can respond to the light, and they can use it to fix CO2, convert into biomass or acetate. And also another example from Julia Buck, also collaborate with our group in Cambridge. So we have carbon dots as a photosensitizer, and we use mesylvelaginine as an electron shuttle, and we deliver electrons into Shivanella, and we can also fix CO2 or convert, for example, pyruvate into lactate or fumarate into succinate. And most recently, we also leverage the collaborations. We do use a click chemistry trying to directly covalent link the photosensitizer to the outside of the membranes and to have a more defined or clearly defined or more efficient electron transfer pathway or as a biohybrid to do this kind of research. So of course, we're going to have more exciting results coming soon. So hopefully in the future, I also have the opportunity to present my research in Cambridge. But with all that, I would like to thank your time, and I'm happy to take any questions or suggestions from the audience. Thank you.
Wonderful talk. Thank you so much. This is brilliant. I also glad to see the collaboration between you and Josh resulting in wonderful work. He actually gave a talk, many months ago in this seminar series. So Karen, do you have any comment or question?
I think it's really very interesting. And I really like electrochemistry as an output. So we've been working too long with fluorescence. So I know optical sensors are not that great. So how easy is it to design ones for new molecules from scratch? Is it similar to how one would put together something that expresses GFP, or is there a bit more difficult?
If you're talking about the electron transfer pathway, it's kind of we already figured out shortly in Caroline's lab. But still we have mutations problems, or we don't have a similar level as when we engineer E. coli and compares to original organisms. So there's still some limits we need to go through. But for sensing, I think especially in this case, because you only need to read out the signal difference, it's already efficient. And I shouldn't say it's easy, but it's very doable or very well studied already. But if you're looking about how we developed, if I can go back the protein sensor, that's basically leverage decades of work. So in this case, actually, we're not or Josh didn't invent this from scratch. We take ideas from literature and use the binding ligand already published, and we know it's working, but with scale, it takes a lot of efforts, especially for Josh. He needs to figure out where will be the best position to split a protein, and you make sure when you split it, it don't have function, but when you somehow merge together, it can rebind together and also restore function. So that also takes a lot of efforts. So yes, so if you're really thinking, if you have one specific target, and if you want to engineer a sensor based on that, that will still take some time of work to do that.
Great, thank you.
Karen, do you have any additional? Okay, so I have one question from audience. If someone asks question, do the environmental samples need to be deoxygenated for the EET sensor to function?
So basically, if I understand correctly, basically we are asking if the samples need to be the oxygens in the samples need to be taken out or not during the sensor. I think that's also one key question that our reviewers ask. So to respond to that, well, hopefully the audience is not a reviewer, because we try to convince the reviewer. Okay, yes, when we do experiment in the lab, we're trying to purge nitrogen and trying to get rid of oxygen, because oxygen is also act as the electron acceptor, which means it's going to interrupt your electron signal. But there's two reasons we think it's still working. One, we do try to working system, like you still have sort of oxygen in the system, you can still work. Of course, the signal is not amazing compared to no oxygen. And two, when you think about in the real world, and you think about a river or ocean, if your sensor point is not just above the liquid surface, if you go further down, actually the oxygen level is not that too high, and you kind of also avoid the oxygen effects from the nature. But again, yes, oxygen should be taken care of, and oxygen could affect a little bit signal. But if you use sensing, you can still have measurable or good enough signal for you to detect the compounds.
Wonderful. I have another question from Maya. With splitting proteins or inducible dimerization, are you doing this by hand? And do you have any plans for ideas of how to do this computationally?
So basically, we're asking how we're making the protein switches, right? So when Josh developed that, he basically had tools that bioinformatics tools that you can use it to run based on the protein structure, and you can find potentially some of the spots that likely can help you to successfully split the protein into a switch. And later, of course, you need to create a library and you need to screen that. And I think what he did basically order a big library from the company, and you have barcode as an insertion there, and you can just randomly insert different locations of the protein. and you test it by function. And after you test it, verify that, you're sequencing back to figure out where is your insert, and you know where could be your engineering point to make it into a switch. So yes, it's not entirely by hand, but it's definitely take a lot of time or efforts to do by now. Hopefully, we can have better ways in the future with the AI or with more robot arms to help us to do the pipetting.
I have one quick, probably challenging question. So I always wonder how could, I enter this field, this but this is still fancy thing to me. The question I asked when I consider, there are some strain of bacteria doing amazing job in terms of electricity or electronic, whatever, transfer. But is there any good way to screen more natural bacteria out there to, use probably with a better ability?
Yes, that's a very good question. And actually, I do have the answer to that question. Because,
wonderful.
Yeah, thank you. So even in our research in Caroline's lab, we use colorimetric assays to detect electron transfer bacteria. So basically, imagine you have a type of nanomaterial going to change color when it get reduced or when they taking electrons. The specific compounds we use is tungsten oxide. So tungsten oxide nanoparticles itself is a grayish color, but when it reduces or get electrons, it will change to a blue color. So now imagine if you have environmental samples, if you can just separate them by colony and grow them up, and you can just mix them with the tungsten oxide and just do the incubation and detecting how much the color being changed. And then you know if the bacteria has the electron transfer pathway or not, or if they have the ability to do the electron transfer. So that's a very fast and easy way to do screening.
That's wonderful. So I may also enter that new area of research in the future in that case. Okay, now I see, 3 minutes left before 11. Let me double check. I don't see any additional question from audience. If Karen does not have any additional question, I want to close and then we start to free chat. Okay. So thank you so much for joining and staying today. We'll meet again next week on October 26th, Thursday, the same time, the same Zoom link. We'll have Professor Doug Densmore at Boston University and Dr. Yu Chang at University of Wisconsin-Madison. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks, I stop recording. Give me 1 second.